to get used to the whole thing. Um, yep. So welcome to the PAL session on biochem, protein structure and protein folding diseases. My name is Abdelrafour Houdane. I think I taught some of you guys in pre-med, and this is kind of like my first year PAL for the year. So obviously super duper excited. Um, just kind of like a little structure of today's talk. It's not going to be very long. This topic is not very hard, in my opinion. It's kind of like very logical, which is nice. We're going to start with a little kind of like introduction to kind of just get the ball rolling, as always. And then we're going to move on to protein structure, the process of protein folding, and when it goes wrong, so protein misfolding. Then we're going to move on to like the clinical side of clinical biochemistry. So a lot of like diseases, and we'll wrap it up with like exam style questions. We're gonna have multiple points where I'm gonna stop and ask you guys if you have any questions, so that way you guys can stay on track. And if you have any like really big questions, you can always ask them towards the end. And I hope you guys find this very informative and fun. So, biochemistry, right? It's been 6 years, maybe, I think, since I did biochemistry, and I don't really remember much, but I do remember distinctly like how much not fun it was. I think most students are kind of like scared of biochemistry to some extent. It is not the easiest topic that you take in first year, but ultimately speaking, I do think if you approach it logically, it is not as bad as it initially feels like. Okay, and obviously, you're gonna figure this out, especially later on. But like proteins are probably the most important thing in like existence in the human body. It really forms the foundation of literally everything. Okay, uh, all the enzymes. It's the juice of life, in my personal opinion. So it is a very important topic, and something that you should be kind of like well aware of. So ultimately, like the first question I think we should always answer is, what are proteins? Uh, Proteins are just like polypeptides, like the name suggests, a string of amino acids joined together. I'm seeing like the chat go up, just kind of making me kind of like scared. Oh, okay. I got worried for a second. So, what are proteins? Proteins are ultimately just polypeptides, right? Like the name suggests, a string of amino acids joined together to make something greater, right? These are proteins. Um, and I think it's very important for us to revisit amino acids, just because, again, it forms the foundation of everything we're going to do from now on. And it's always important to remember, like, all amino acids exist as a template. So they are all identical with one like key difference. So the template of an amino acid, you have a central carbon atom, and then you have it's bound to four different molecules, right? It's bound to a hydrogen atom. It's bound to something called the carboxyl group, which is just carbon, double bond, oxygen, and OH, and an amine group, which is going to be nitrogen and two hydrogens. Then you have something called a variable group. Now, this variable group is what distinguishes different amino acids from each other. Each amino acid is different because of the variable group. So this template of amino acids exists for every single amino acid out there. And for example, let's take valine. It looks scary when you look at it, but again, if we break it down, we will notice that the template is always going to be true. So you have a central carbon, you have an amine group towards the left, that's the N and the two hydrogens. You have the carboxyl group towards the right. Then you have that kind of like turquoise thing on top. That is your variable group. That's what makes valine valine. Now, the reason why I am like emphasizing the importance of knowing the template is because it will allow us to answer my next question, and is, how do we join two amino acids together? We said a protein is a collection or a string of amino acids. So how do I join them and link them together? The true answer to that is by joining the carboxyl group of an amino acid with the amine group of another. And if we go back 2 slides, the amine group is the NH2 bound to every single amino acid, and the carboxyl group is going to be the one with the double bond oxygen. So if I want to join two amino acids together, I join the carboxyl group of one and the amine group of another. I'm not very much a very wordsy guy. I need to see images. So the best way to demonstrate this is by seeing it take place. And we call this process, by the way, something called a condensation reaction. or a dehydration reaction. Why? Because in order for us to join those two together, I am removing a molecule of water from the two amino acids. So let's say, for example, I want to join two molecules of valine together. I will take the hydroxyl group from the valine and the other hydrogen from the other valine, and we will remove a molecule of water. So I'm combining the carboxyl group of valine 1 with the amide group of valine 2, and together, boom, we get a condensation reaction. Now, I'm going to be honest with you, this schematic looks really ugly. I'm not the best in like making fancy diagrams. So I will show you guys how it really looks later on. But again, the peptide bond is the one that I highlighted in pink. So again, imagine I want to combine a glycine molecule with an alanine molecule. I will basically, again, take a carboxyl group of glycine with the amide group of alanine, join them together, and then we have the formation of a peptide bond. That is going to be the bond between the carbon and the nitrogen of the other. And again, this is called the condensation reaction. Again, another example, phenylalanine and leucine, same thing. I take the carboxyl group of phenylalanine and the amide group of leucine, remove a molecule of water and join them together to form a peptide bond. And this is, at its simplest form, how we join two amino acids together. Now, the whole purpose of this topic is to go over the different levels of protein structure and then to understand the pathology that is associated with it. And we're going to go over 4 different tiers or levels of structure. That is going to be the primary structure, the secondary structure, the tertiary structure, and finally, the quaternary structure. So does anyone have any questions at this stage?
No.
Okay. So if there's no questions, we can move on to the next part. And that is... Okay. So now we're about to dive into protein structure, right? Now, the best way to kind of understand and to stay oriented for me personally is to always think about things in terms of bonds. At each level within the protein structure, there's different bonds that are being formed. And it is the different bonds that kind of like dictates the different structure that we have. And they will make more sense when we go through different examples, but just kind of always think of these increasing levels of complexity in the design of the protein as a direct result of different bonds being formed. So let's start with the primary structure, which is obviously the simplest, and we already kind of went through the bulk already. So the primary structure of a protein is the simplest level of protein organization. It is simply a string of amino acids joined together by those peptide bonds. And again, how do we form peptide bonds? It is a condensation reaction. We're taking a carboxyl group of one amino acid, an amide group of another, and joining them together. This is the primary structure. So here's an example of insulin, right? You can see it's a long chain of amino acids. We have glycine joined with isoleucine, joined to valine, joined to like glutamic acid, joined to like glutamine, cysteine, etc. It's a string of amino acids. This is the primary structure. It is the simplest level of protein structure. Now, the exact order of amino acids is going to be determined by your genetic code. It is determined by the DNA that will kind of tell you exactly why is it isoleucine before leucine, etc. Then we move on to the next level, which is the secondary structure, right? But before I dive into, what is the secondary structure, etc., I want to revisit that primary sequence and reintroduce 2 new terms, the polypeptide or the peptide backbone and the side chains. Now, the best way to understand this is to use an example. So imagine we took our good old trusty valine and we have basically 4 valine molecules together. Obviously, I did not like remove the hydrogens and stuff. So like this is technically before condensation reaction, but just for the sake of like simplicity, here's your 4 valine molecules, okay? Imagine, why did this? Yeah. So imagine now we look at purely all those carbons and all that thing. This is called your peptide backbone. It is the collection of all the carboxyl groups, the amine groups, and the central carbons found within your primary structure. So this is your peptide backbone, also known as your polypeptide backbone. Then you have on top of it the side chains. The side chains are the variable groups within the amino acid. And that's going to be different based on the amino acid itself, right? And the reason why I'm bringing this up, the reason why I'm stressing all of this is because it ties into what the secondary structure is. The secondary structure is what happens when we have the formation of hydrogen bonds between this peptide backbone. And when we have the formation of hydrogen bonds, this leads to the formation of something called coils and folds. By definition, we call these coils alpha helices, and we call the folds beta pleated sheets. And the best way to kind of like really visualize this mentally is to look at it visually through examples. This is how an alpha helix looks like. It's a coil. Think of it like, I don't know, curly hair to some extent. And the beta sheets are kind of like pleated. They kind of folded into each other. Now, let's take, for example, again, revisiting the simplest structure of a protein, which is going to be your primary structure, your string of amino acids. And again, it looks really overwhelming. It's confusing. It's a bunch of shit going on. But again, let's break it down to the simplest form. It's the same template, a central carbon with an amine group to the left, a carboxyl group to the right, a hydrogen group, and a variable group. And that is true throughout. It's the same repeating structure. Then we have the peptide bonds, right? Which is the joining of the carboxyl group and an amine group from one amino acid to the other. And again, previously I mentioned the peptide backbone. And the reason why it's so important is that because this is when the secondary structure comes into play. In the secondary structure, like I previously mentioned, it is the formation of hydrogen bonds within this backbone. So that's exactly what is happening. Here you have the formation of a hydrogen bond between the carboxyl group of amino acid number 1 and amino acid 4's amide group. And when you form a hydrogen bond between these two amino acids, which are considerably further apart, you form a kink. That hydrogen bond will visually and like structurally create a kink within the amino acid frame. And that kink will lead to the formation of an alpha helix. Now, it's hard to kind of like explain this without seeing like a 3D representation or a model, unless you're really good at visualization. Me personally, I'm probably not the best. So there are some nice videos on YouTube that kind of show you how this happens in real life. But think about it. If you have the formation of a bond from two amino acids which are further apart, that will cause a kink and it will begin to coil into each other. Now, this is how we form that alpha helix. So an alpha helix is simply a hydrogen bond between carbonyl or carboxyl group of one amino acid and the amide group of another. And all of this is happening, and I really want to stress, within the peptide backbone. So one important thing to also add on is that the formation of these hydrogen bonds are going to be parallel to the backbone. Now, because again, each primary sequence is going to be different. Each amino acid is going to be different. We usually don't tend to notice alpha helices being formed when the side chains, when those variable groups are very bulky, or if they contain glycine, because these are like not very mechanically constructive to the formation of an alpha helix. Honestly, I do believe that this is very, very low yield information. I obviously do not remember ever seeing this later on in like my medical career kind of, but it's on the slides and I just wanted to add it for the sake of completion. Now, some good examples of where we can find alpha helices in real life. That's going to be in a group of proteins called keratins, which is what we find in hair, what we find in nails, and also myoglobins, which is what we find in like muscles and stuff, like cool stuff, basically. Now, beta pleated sheets, it's the same concept. It's the formation of hydrogen bonds, again, within the peptide like backbone. But the main difference between a beta pleated sheet and an alpha helix is the direction of these bonds. In an alpha helix, we said that they are parallel, right? They are parallel to the backbone. However, in beta sheets, they are basically perpendicular. So that's how we form the structure. Now, proteins in general, they kind of like can have varying degrees of alpha helices and varying degrees of beta sheets. Stuff like myoglobin, like I said previously, are mainly composed purely of alpha helices. Antibodies are made purely of beta sheets. Then you have stuff which kind of exists in between, like flavodoxin, which is like a combination of alpha helices and beta sheets. But these are just examples of how these can present in real life. So that kind of like wraps up the primary structure and now wraps up the secondary structure. Any questions so far?
Oh yes, I have a question. What does mean kink? It's bond
Uh,
between two amino acids, right? That's it.
yeah. So a kink, I'm trying to think of a good way to describe the word. Do you know how like it's like a bend? Think of it like a piece of paper, and then suddenly you bend it from the inside. It will create like an internal kink. The best way I would say, either if you can watch a video, you will see like how it happens. Salaha, I don't know how to say it in Arabic. If anyone knows how to say kink in Arabic, that would be great. I don't. But the best way how to describe it is like it bends from inside, and because it
Oh,
bends from inside, it will turn into itself. You know.
yeah,
Uh,
okay, got it. Thank
so
you.
yeah. Anytime. But guys, I strongly recommend you guys to watch a video because like when you see it in 3D, it makes so much sense.
I have another question.
Yeah, tell me.
Can we go to the side chain and the backbone slide?
Uh, Tell me when to stop. Yeah,
Fourth, yes. Down.
this
Slide number 40. Slide number
40.
40. Yes, we said that this is the backbone. We said that the side chains are the R groups, right?
Yes. Exactly.
So so why the O attached to the carbon isn't like part of the backbone, the down
Very,
one?
very, very, very good question. And you are absolutely right. That oxygen is part of the backbone. Salaha, I just kind of like was lazy and I did not like highlight it into orange, but you are absolutely correct. The carboxyl group are part of the backbone. Very, very good point. Salaha is just I did not kind of like visually represent it, but it is part of that backbone. The side chains, the R1, R2, they are separate. But the oxygen and the hydrogen that is kind of like is there like an annotate tool? Can you see my cursor?
Yes.
This oxygen group here and this hydrogen group here are part of the backbone. It's just because I
Yes.
did not have the time to like extend the orange thingy. But very, very good point.
Okay. Thank
Thank
you.
you so much. Any other questions? Okay, so moving on now to the tertiary structure. So we went through primary structure, which was the chain of amino acids. Then for secondary structure, it was the formation of these hydrogen bonds within the backbone. Now we move on to the third thing, which is very simple. It's the formation of a 3D structure due to interactions between the variable groups. So secondary structure was all about the backbone. Now we move on to the variable groups. And because the variable groups are many, those side chains are many, we can have a bunch of different types of bonds that can be formed. So we can form the formation, we can have other hydrogen bonds, but these times being formed between the variable groups. We could have the formation of ionic bonds. We could have the formation of disulfide bridges when you have two sulfur molecules. These are very, very strong. Or we can also have hydrophobic interactions. Um, Together, these different types of bonds will create that final like 3D structure of your protein. Now, I'm going to revisit the previous diagram and you will notice it's a bit different because initially I wanted you guys to focus on the primary sequence. Now I'm going to introduce to you how the insulin molecule gets its final shape. The cysteine molecule, the cysteine amino acid has sulfur molecules in its variable group. So we form these disulfide bridges. Okay, this is an example of a tertiary structure. It is the interactions between these variable groups. And they will kind of take our secondary structure, our alpha helices and beta plated sheets, and turn it into something even more complex, even more binding internally. And then finally, you have the quaternary structure, which is basically going to be multiple polypeptide chains joining together to form like a super mega structure. So you have a polypeptide chain, which was initially a primary structure, right? A sequence of amino acids with peptide bonds. Then we had the formation of hydrogen bonds within the backbone. So we have the formation of alpha helices and beta sheets. Then we had the variable groups interacting. So we had the tertiary structure. And then if I take one tertiary structure, I take another tertiary structure, and I join them together, I have the quaternary structure. So by convention, the amount of chains we have will change the terminology we use. So if we have 2 chains, we call it a dimer. If we have 3 chains, we call it a trimer. If we have 4 chains, we call it a tetramer, etc. And the best example of a quaternary structure is the good old hemoglobin molecule. If I can just, it's a bit lagging for a bit. But the hemoglobin molecule, as you know, is a collection of 4 different chains of proteins joined together, right? Two alpha chains and 2 beta chains. This is a good example of a quaternary structure. It is multiple polypeptide chains joined together. Let me just go back. Yes, so this is a great example of it all coming together. You have your primary structure there, which is your sequence of amino acids. Then you have the formation of these hydrogen bonds within the backbone, within the peptide backbone, forming your secondary structure. Then the tertiary structure takes place. And then when we join them all together, we get a quaternary structure. And this is just a recap of everything that we've done so far. Any questions at this point? Go for it.
The doctor mentioned something in the lecture. He said that myoglobin is made of one chain, and it's even though it's made of one chain, it's called a tertiary structure. So they can get us with this.
Yes. So myoglobin is different from hemoglobin, right? So so hemoglobin is your classic example of quaternary structure. It has multiple polypeptide chains, but myoglobin is different. It has a singular chain. And the maximum level you can reach when you have one chain is tertiary. So myoglobin will have a tertiary structure, true. But hemoglobin, because it's more than one chain joined together, then you get the option of becoming this super mega structure, which is your quaternary structure. But that's a very, very good point. Thank you for bringing that up. Any other questions? Oh, no, not at all. If you ask me, this details are very, very little, and 100% you don't need to know the position of to be honest, I don't think anyone knows on planet Earth knows the exact position by heart. It's just they know it obviously by biochemistry and stuff, but no, you don't need to know the exact positioning. Just know, I guess, and I don't think even you need to know that, but just know the existence. I just use this as an example, Salaha. But later on, I think maybe when you guys do endocrinology, you may go through the structure of insulin, or maybe towards biochem too. Then you're supposed to know that disulfide bridges exist. That's all you need to know. But the exact positioning, no. That's way, 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 way outside your scope. You guys want to be doctors, not biochemists, ultimately. Any other questions so far? Okay. So moving on to protein folding. So as we said later on, the whole purpose, the whole point of going through the primary structure and the secondary structure and the tertiary is to get this concept that this is how we get functional proteins. A folded protein ultimately is a functional protein. It is able to do its function within the body. However, the process of folding is an active process. It doesn't happen randomly. It happens by a process. And it's really mainly aided by something called chaperone proteins. Chaperone proteins are proteins themselves that's main function is to aid in the process of folding. So what these chaperones do, they promote protein folding, and they also are there to prevent misfolding. And we're going to have a whole section where we talk about misfolding because it's kind of important. So let's go over how chaperones help in that. So let's say I have a polypeptide and it's unfolded. It's not functional. These chaperone proteins, with the function of with the help of ATP, so it's an active process, it requires ATP, will basically bind with our unfolded polypeptide. And then it will basically facilitate the folding process as well as protect it from basically getting misfolded, protect it from maybe like metals or toxins. If they bind, they can affect the folding process. And then they basically disassociate. Now this polypeptide is folded, it's ready to go out into the world and do its job and do its like function. They also want you to kind of understand this whole concept of melting temperature. The melting temperature is simply the degree or the temperature at which 50% of the protein is unfolded. Now, the main concept here is if I take a normal protein and I put it under stress, either, for example, I increase the temperature really, really high, or I put it in some like insane acid or some insane alkaline material, I can begin to unfold this protein. And when I unfold this protein, it is not able to do its job. Now, most proteins unfold around 40 to 60 degrees Celsius, right? This is when things stop working. And you guys are fresh from high school, so I guess you guys remember the official term, which was denaturation. Denaturation is like the unfolding and disruption of the secondary and tertiary structure of a protein, secondary to heat or secondary to like high pH or low pH, etc. And effectively what this is trying to tell you guys that if I put a protein through a lot of stress, if I put the protein through high temperatures, it will unfold. And it is the process of unfolding that kind of like causes it to stop working. It destroys the function of the protein. So the denaturation process usually is irreversible. Once you kind of like screwed up the protein to that point, it loses its function and is not able to work anymore. Just kind of like very simple things. And again, we can do this by changing the pH, we can do this by changing the temperature, etc. So again, this part is like not very hard. I doubt there's going to be many questions, but again, if anyone has any questions, just let me know. Okay, so I'm just going to move on. Protein misfolding. Ultimately, like you guys came here, you guys like joined medicine to become doctors, right? So all this biochemistry is all fun and games. It is whatever, but ultimately you want to understand disease, and that's the whole point of biochemistry. So it's important to note that nothing is perfect in life. Things happen, things go wrong, and that's also true from everything that we've talked about so far. So the big question is, what happens if a protein misfolds, and like should we even care? Now, it is very important to know that when a protein misfolds, number one, it cannot do its job. And we've spoken about this before, right? The whole concept of denaturation, of like melting down proteins, is that when it loses its normal folded shape, it is not able to do its job. But there's another thing that can happen. Misfolded proteins can aggregate. They can stick together. When a protein is misfolded, it begins to act abnormally in a weird way, and it begins to stick with other misfolded proteins. And that's really not a good thing, because as proteins begin to stick together, as they begin to aggregate, they become very, very stable. You know, like the whole kind of like monkey together strong is the same concept. You have one broken protein sticking with another broken protein. When that happens, the proteins begin to accumulate and it becomes too big to break down within the cell, too big to transport. And as it accumulates, it becomes toxic. It becomes toxic to the cells. And when things are toxic, they lead to disease and pathology. So that's the final end outcomes of when proteins begin to misfold. So what causes proteins to misfold? See it as three main things. Number one, DNA mutations. If for whatever reason I have a mutation in my DNA, the protein itself can form weirdly and abnormally, and they basically are not able to fold properly in the first place. Then you have the stresses, high temperature, the pH, all that kind of like stuff, or even just like the continuous motion of life. Sorry about that. Just continuously as time goes on, this protein undergoes a lot of stress doing its normal day-to-day life that it can eventually spontaneously misfold. And finally, you have all the environmental stuff. This is stuff like toxins, metals, pesticides, all that stuff can go to the protein and cause it to misfold. And we know that ultimately a lot of our proteins are going to misfold. So we need a system in place within our body to deal with those misfolded proteins before they begin to accumulate. And the system that we have in place is something called the ubiquitin proteasome system. The way how it works is this. If a protein begins to get misfolded, the body is able to detect that, and it labels and tags it with something called ubiquitin. So you have your misfolded protein, and you basically mark it with this ubiquitin. This marker basically tells the body, hey, this protein needs to be broken down. And we do it by another protein called a proteasome. Now, from the name proteasome, it is a protein that breaks down other proteins. But we don't want a proteasome to go haywire and start breaking down all the proteins in the body. So this proteasome only breaks down proteins that are tagged by ubiquitin. So you have a protein, for whatever reason, it misfolds. This is detected by the body and they tag it with ubiquitin. Now the proteasome is able to break it down and prevent it from accumulating. This is the whole concept of the ubiquitin-dependent protein catabolism, or the ubiquitin proteasome pathway. It is our body's system to prevent protein aggregation and to prevent protein accumulation. However, if for whatever reason you have more misfolded proteins that your body can break down, you will eventually get disease. And this is going to tie us in to the next kind of like aspect where we go into the diseases related to protein misfolding. So before we dive into clinical biochemistry, does anyone have any questions so far from anything that we've taken?
I just want to.
Yep.
If the stress factor, like psychological one, can like also be included in the factors that affect protein folding, the psychological one.
I think what they mean here more about like mechanical stresses or physiological stresses more than like psychiatric stresses, you know. They mean stresses more on like a biochemistry level, on a molecular level. So like for example, interacting with oxygen over time, you'll later on be introduced to something called reactive oxygen species. These can kind of like create environment like stresses, but like biochemical stresses. Not really kind of like mental stresses. That's more of a separate topic, kind of.
Okay, thank you.
Good question. Thank you so much for asking. Yeah. Sorry, guys. I'm so the ubiquitin-dependent protein catabolism. I'm gonna just repeat that real quick. So as I was saying, the whole kind of like concept here is we need a system within the body to allow us to identify and break down those misfolded proteins. But at the same time, I do not want to have something that is, because ultimately, like misfolded proteins and normal proteins are still proteins, and proteasomes break down proteins. So I want a system in place where I only break down the misfolded proteins. So let's say, for example, I have protein number 235, random protein floating in my cell, right? And for some reason or another, maybe it found a pesticide or a toxin and it underwent spontaneous misfolding. That is not good. I do not want misfolded proteins within my body. So what happens is the body will detect this, will be like, yo, hold on a second. This protein does not look normal. Tag it with ubiquitin. Now, ubiquitin is a tagger. Its job is just to say, hey, this protein needs to be broken down. Please break it down. So what happens is we basically tag this misfolded protein with ubiquitin. Now, proteasomes only break down and recycle, right? Ubiquitin tagged proteins, because we do not want to keep breaking down random proteins within the body. So that misfolded protein, which is now tagged by ubiquitin because it's not normal, is going to be detected by the body, and proteasomes are going to break it down. Because we know if we do not break down those misfolded proteins, they are going to begin to accumulate. If they begin to accumulate and accumulate and accumulate, eventually they're going to be toxic and the whole cell is going to die. So the ubiquitin system kind of works in two ways. Step number one, you want to tag your protein by binding it with ubiquitin, which will mark it as, hey, it's abnormal. Then you have your proteasome, and your proteasome will then break down that ubiquitin targeted protein, which is misfolded, and you break it down into those little peptides, and then they can basically be recycled by the body to make a normal functional protein. Does that kind of explain the ubiquitin pathway? Perfect. Now, to answer the question about chaperone proteins, so the whole process of like protein folding, it's an active process, and a chaperone is someone that helps someone, right? The whole concept here, it's a protein that's job is to help other proteins reach their final form, to help these proteins become properly folded so they can do their job. And the way how they work is they facilitate protein folding and they prevent them from getting misfolded. Now, the exact mechanism of how it works and the deep biochemistry I feel way outside the scope of what you guys are doing. Yes, if you guys are interested, you can search and discover there's many ways of how chaperone proteins kind of like facilitate the formation of those bonds. Because ultimately speaking, the conversion of unfolded to folded is all about the formation of bonds, right? The formation of hydrogen bonds within the polypeptide chain, the formations of the bonds between the variable groups, and the chaperones help in that. They help in those formation of those bonds. But the general big picture stuff, because I don't want to overwhelm you with information that's maybe not as high yield, is just understand that these chaperone proteins work by helping the proteins that are unfolded become folded. They are kind of like wingmen to a certain extent. Does that kind of like make sense? Okay, so guys, this is, for me personally, where the fun part begins, which is clinical biochemistry. Because ultimately speaking, like I said, you guys joined medicine for a reason. Now, we said previously before that the primary structure of a protein is determined by the genetic code. So what happens if you have a mutation? The best way to kind of explore this topic is by exploring and going through different diseases and seeing how mutations can affect the functional outcome. And sorry if there's so much background noise, by the way. Let's begin with something called cystic fibrosis, which you guys may have taken in high school, which you guys are going to take a lot of medicine because it's a very high yield disease. All of us have something called a cystic fibrosis transmembrane conductance regulator protein. Name is too long, so we just call it CFTR. The function of the CFTR protein is to basically pump chloride ions into our airways and other stuff. And that's very important. It's important for the normal function and the normal structure of the human body. However, imagine if someone has a mutation. And the most common mutation that happens in cystic fibrosis is something called delta F508. Delta F508. What happens here is delta just means deletion. So deletion, it's a deletion of a single amino acid. The amino acid phenylalanine at position 508. So delta F508 just is supposed to tell you it's a deletion of phenylalanine at position 508. So what happens is a person gets a deletion. That will completely alter the primary structure of their amino acid, because that's a sequence of amino acids. But now phenylalanine is missing. So instead of isoleucine, phenylalanine, glycine, suddenly it's isoleucine, glycine. And it may not seem that significant. It's a deletion of literally a single amino acid, but that has insane implications in everything else. Because if I am affecting the primary protein structure sequence, I am effectively going to affect the secondary structure, right? Because the secondary structure is dependent on the peptide backbone. So if I alter the peptide backbone and I alter now the variable side chain, eventually the final protein is going to be different. It is not going to be the same as before. And that means the function of the protein is affected. So in these patients who have this abnormal CFTR protein, they are not able to pump those chloride ions outside into the lumen of the membranes. And what happens is all that mucus within the airways begins to accumulate. And a lot of things go wrong from then on. These patients have problems, they get repeated infections. They have problems with their pancreas because their pancreatic ducts get filled up with mucus. So they begin to have failure to thrive. They're not able to grow very well. They get repeated infections. And their entire respiratory tract fails to function normally. And all that is due to a single mutation at the level of the DNA. So this is a good example of how a DNA mutation can lead to an abnormal protein, which then leads to disease. So that's one example, cystic fibrosis. Another example is sickle cell anemia. Again, this is the structure of hemoglobin. And hemoglobin is a quaternary structure because you have multiple polypeptide chains joined together to make one megastructure, okay? Now, imagine if a patient has a single, not now a amino acid, a point mutation at the level of DNA. So instead of it being CTG, it becomes being GTG, it becomes GAG. And this leads to the substitution of glutamic acid to valine on the level of the primary chain, in the level of the primary structure. So in cystic fibrosis, we had a deletion, right? We had phenylalanine going missing. Here, no, nothing is being deleted. Just a single amino acid is being substituted for another amino acid because of a very small mutation on the DNA. And as small as it sounds, as insignificant as it may be, this very tiny mutation has insane implications on the patient because what happens is you have altered the primary sequence of the amino acid. The conversion of glutamic acid to valine has a big consequence because glutamic acid has a acidic side chain, but valine has a non-polar side chain. So what happens when you substitute glutamic acid to valine is that you completely alter your secondary and your tertiary structures. You begin to create these hydrophobic regions that begin to stick together and agglutinate. And what happens to the red blood cell when the patient has low levels of oxygen within their body, the entire cells begin to become deformed, and they form these very characteristic sickle shapes. And that leads to the manifestation of sickle cell, which is going to be these pain crises. They will have anemia. Their hemoglobins are going to be low. Their body is going to be destroying those RBCs because they get stuck within the blood vessels. It's a very, very nasty disease. And I know you guys are still first years, but once you start rotating in hospitals, you will notice sickle cell is very, very common in Saudi. And especially when you do your ER rotations, you are going to be facing these patients who are in extreme pain. And all of that is from a single substitution of one amino acid to another. But that has huge implications, both from the level of the primary structure to the secondary structure to the quaternary structure itself. And the reason why I'm bringing these examples is because I want to demonstrate the importance of the biochemistry in real life. Because ultimately, this is what you guys are going to be dealing with as future doctors, inshallah. So that was good examples of how DNA mutations impact the final outcome of your protein. Now I'm going to shift gears and move on to misfolding and aggregation, etc. And a good example of that is something called Huntington's disease. Huntington's disease is a neuro disease. these patients are going to present with these tremors. They're going to present with like abnormalities in thinking. They're going to start having like memory loss, abnormal behavior. Their personality begins to change, and it's a genetic disease. And the underlying pathogenesis of Huntington's, it is something called a polyglutamine repeat. Now, we all have within the Huntington protein a certain amount of base repeats. It is the CAG, but most of us have it less than 27 times. It is repeated within our primary sequence, right? In patients with Huntington, they have a mutation, and the amount of CAG increases. As I said, usually is less than 27. They have it much, much more. And because they have so much of these, they lead to a mutated protein. Now, this mutated Huntington protein loves to accumulate. It is abnormal. Previously, I mentioned different ways a protein can misfold. And one of them was DNA mutations. This is a good example. You have a DNA mutation, a polyglutamine repeat that leads to the formation of an abnormal protein that loves to accumulate. It is misfolded. And when it accumulates, it accumulates more than the ubiquitin proteasome pathway can break it down. Then it becomes toxic, but it only accumulates within the neurons. So the neurons begin to die, and these patients begin to have like brain-related symptoms, memory loss, abnormal behavior, abnormal movements, etc. So that's an example of how misfolded proteins from a mutation can lead to disease or pathology. Just take a little water break, 1 second. Finally, we have other diseases known as amyloid diseases, right? Amyloid is a bit of a weird name. I've never heard of it before, so let's go through it. Amyloids are simply protein aggregates. We call these protein aggregates amyloids. And again, you have here a problem in the breakdown, or they begin to accumulate, and it is the accumulation of those protein aggregates that cause disease because they are ultimately toxic. As they begin to accumulate and accumulate and accumulate, they break down body. They begin to destroy those cells. And like I said, there are many different types of like conditions that are caused by these amyloids. I'm going to go over 3 today. One of them is Alzheimer's disease. One of them is Parkinson's. And one of them is just called amyloidosis. Alzheimer's, I think a lot of people know this already. It's a very common disease of like memory, right? Old people suddenly have memory problems. They begin to forget basic stuff. They begin to forget how to drive. They start forgetting their neighborhood. Eventually, they start forgetting people. It's a very heartbreaking disease. Now, we all have a protein in our body called the amyloid protein precursor, or the APP protein. Now, our body has a way of breaking it down. It has it has an enzyme called the alpha secretase, and the alpha secretase cleaves it right in the middle, and it breaks it down, and it's able to be broken down by the rest of the body. And I've shown multiple images of the same thing. It is a big protein that is broken down by alpha secretase, and it's broken down into smaller components, which is good. However, some patients have a problem in this breaking down. And it's kind of like cleaves the enzyme in a weird way. And when it cleaves the enzyme in a weird way, it leaves behind this kind of like fragment, that fragment in red, which is called an A beta peptide. Now, the problem with A beta peptides is it absolutely adores other alpha beta peptides. And they begin to join together, they begin to accumulate within the brain and form these amyloid plaques. And these amyloid plaques, right? These amyloid plaques are neurotoxic, and they are associated with the problems that eventually lead to Alzheimer's dementia. So this abnormal breakdown of protein is usually due to mutations in the enzymes that break down APP. So we all have amyloid precursor protein, and we all are able to break it down. However, some people have mutations in the breakdown process, which leads to accumulation of this alpha beta peptide. So that's one example of how these accumulation can lead to disease. In this case, which is Alzheimer's dementia. So it is the accumulation of these alpha beta platelet sheets, which are insoluble, which then cause problems within the brain. In Parkinson's disease, a very similar kind of like process, but here you have accumulation of something called alpha-synuclein. This accumulation of alpha-synuclein, it is insoluble. It forms these kind of like fibrils, these beta-pleated sheets that accumulate within the neurons. And again, they basically break down. They are neurotoxic. They break down the neurons, and these patients begin to have Parkinson's. Now, I don't know how much of if you guys are into sports, but like the late Muhammad Ali was someone who had Parkinson's, who had those kind of like tremors and abnormal movement and that very characteristic shuffling gait. That's all part of Parkinson's. And it's something that you guys are going to study in your second year neurology rotations. And you're going to find this out. But the central pathogenesis of this condition, again, it's the accumulation of these abnormal proteins, these alpha-synucleins within the neurons. Finally, again, very low yield, but again, for the sake of completion, we all have something called transthyretin amyloid, right? But our body is again able to break it down. However, some people are not able to do so. So these transthyretin, which we all have, begin to accumulate, and then they begin to accumulate in different organs like the heart or the nerves. And then we begin to have disease like restrictive cardiomyopathy. The heart is not able to relax properly and they go into heart failure, or they begin to have nerve problems. Again, the only thing I want you to know from this slide is the idea, is the concept that if you have an abnormal misfolded protein that begins to accumulate, it can lead to disease and it can lead to pathology. And it was a lot of these same examples that are repeated just to kind of really drill this message home and just to kind of tell you why we focus so much on learning about the primary structure and the secondary structure and the tertiary structure. Because me personally, I don't like the idea of like focusing a lot on biochemistry and not knowing why. I remember, especially in first year, I would be like, why am I learning this? I don't care about biochemistry, right? I came here to learn about medicine. So this is kind of like my way of telling you, hey, what you guys are studying are kind of important for a bit. Then we have the last disease and the end of this topic, which is prion diseases. Does anyone have any questions, by the way, so far? Okay, so no questions. I'm just going to dive real quick. Okay. Could you repeat Alzheimer's and Parkinson's? Sure. I'll be honest with you guys. The exact pathogenesis of like Alzheimer's, it's something you're going to be taking later on. I added these slides just really more for the purpose of completion. But the whole concept here is just to give you an idea of the relationship between accumulation of proteins and these pathology. So in Alzheimer's, the central like pathogenesis, the main mechanism is you have abnormal secretase activity. And the role of secretase is to break down that alpha beta peptide. But when you have a problem in breaking it down, it begins to accumulate. And when it accumulates, it forms these amyloid plaques within the brain, which is associated with Alzheimer's. And similarly, in patients with Parkinson's, it's an accumulation of alpha-synuclein. Just know the general idea that in Parkinson's and Alzheimer's, you have accumulations of proteins, of these misfolded proteins that cause pathology and cause disease. Going down into the nitty-gritty of how, sorry, why is it lagging? Going into the exact way how Alzheimer's is caused and how Parkinson's is caused, honestly, this is something you're going to be taking in second and third year. I don't want you guys to focus on this now because it's outside the scope of biochemistry. You already have way too much information to remember. So just know that it is the accumulation of these alpha beta pleated sheets in Alzheimer's, and it's the accumulation of alpha-synuclein in Parkinson's. I'm going to quickly wrap up prion diseases very, very quick. Now, what is a prion? A prion is just an infectious protein. Now, this sounds very, very weird, right? How can a protein be infectious? And honestly, for me, prions are probably the scariest disease known to man, just because I'm going to explain why. But the way how prions work, it's an abnormally folded protein, but it's infectious. If it meets with a normal protein, it can cause it to also misfold. So you start with one abnormally folded protein that creates this chain reaction. This chain reactions of multiple misfolded proteins, one after the other. And that is really, really bad because that means you can start with one abnormally folded protein and get a disease from the get-go. Usually these diseases cause neural damage, they cause brain damage, and the umbrella term for all those conditions is called transmissible spongiform encephalopathy. Transmissible is basically telling you you can spread. It can spread from one person to another. It can spread from like animals to humans. Spongiform to do with the brain, and encephalopathy because it causes literal brain damage. And I don't know if you guys remember, you guys probably were like very, very young, but there was this one period in time when, especially in the UK, we had bovine spongiform encephalopathy or mad cow disease. It's the main reason why people who are born in the UK at a certain period in time are still not able to donate blood, and why the UK cannot give blood products anywhere, because we had an epidemic of this within our population. So the main idea was it affected cattle, and then humans ate infected cattle, right? And got infected with these prions. And then eventually with time, they developed something called Creutzfeldt-Jacobs disease, which is where these prions go to the brain and completely destroy the brain. And people would like be fine, they'll be normal. Then suddenly within a year, they'll be dead. They'll have insane gradual decline. They'll go from just normal adults to having problems with memory, problems being able to walk, problems with literally everything. And then they're usually dead within a year. And because it's transmissible, people who, for example, took blood product from someone who was infected, people who are working with human bodies, or who worked with like transplants, or who ate infected cattle, all were at risk of developing this very, very dangerous prion-related disease. And the whole, again, concept of these prion diseases is that it's an abnormally folded protein that can cause other proteins to misfold, and it becomes this very, very nasty, nasty chain reaction of the body. Can you name the pathogens mean time? Sorry, I didn't understand the question, Mubarak. What do you mean?
Can you name the main types of the pathogens, like the three types you named?
You mean under the amyloid category?
Yeah.
Yeah. So it's Alzheimer's dementia or Alzheimer's disease, right? Parkinson's, which is the next one. So this is Parkinson's disease, and this is just known as amyloidosis.
No, what I meant was like the ones with the DNA problems, and the one with the misfolding, and the one with the what was
okay.
that?
So we had cystic fibrosis, which was the first one, right? The
Yes.
mutation in the CFTR gene, CFTR protein. So that's cystic fibrosis. Then we had sickle cell. And in sickle cell, it is not a deletion of an amino acid, it is a substitution of amino acid. We go from glutamic acid to valine. So that's sickle cell anemia. Then we have Huntington. And in Huntington, we have polyglutamine increase or a polyglutamine repeat. So instead of having less than 27, we have more than 35. And when we have more than 35, the protein itself becomes deformed, begins to accumulate, and lead to neuronal cell death. So these patients would present with memory loss, with these abnormal body movements, and they will have positive family history. So you ask them, hey, do you have any family members who have something similar? And they'll be like, yeah, my dad died when he was 56. My granddad died when he was 63, and they all had the same thing. So you know it's an inherited condition. It's a genetic disease. Does that kind of like answer your question?
Yeah, thank you.
Anytime. Any questions so far from this section, guys?
I don't have a question from the section, like I have a question in general. Can you study from this PPT, like rather than the doctor's slides?
I mean, I hope so, right? Like I made it based on his slides. So inshallah, I hope like it's the same content mainly, just like restructured. But obviously, like ultimately, listen, I'm not the one who's studying your exam. I don't know if you're willing to take that risk. Salaha, if you feel like if you're getting all the questions, when you start doing practice questions, if you feel like you're getting all the questions right, then you know the topic, right? The way how like I kind of like studied during like my time in Al Faisal was if I'm getting questions right during the exam, like the exam questions and the repeated questions, then I know, okay, I understand the topic. I can move on. Plus, you guys have one very good opportunity, which is the midterm, right? Midterm is what, Tuesday?
Yes.
Yeah. So the beauty of midterms, it kind of tells you what areas are you good at, what areas do you need to focus in. I mean, I made these slides kind of like يعني with the ultimate goal that it's everything you need for the exam, you know. بصراحة like things change year within year, and bro, like I'll be honest with you, when I did biochem, it was 2019 or 2018. It's been what, like a long time ago. So I don't know like how questions changed. We have, by the way, before I like we end the slide, we have some questions, which is obviously a great way to test your knowledge. So let's do that now. Anyone wants to read? Okay, so I'll just read it. I'll read it. I think that's better. So you have a 56-year-old pathologist was taken to his family doctor by his son. He was showing mood changes, minor loss of memory, and decreased motor skills. During the patient history, it became clear that over the course of his career, he had on occasion cut himself using the instruments he had been using on the cadavers he had been working on. A potential explanation for his symptoms is abnormal aggregation of which of the following proteins? Is it A, hemoglobins in the RBC? Is it B, fibrillin in the extracellular brain compartment? Is it C, a truncated neural protein? Or is it D, a misfolded form of a normal protein? Okay, we got a bunch of these. Okay, seems like a unanimous choice for D. Interesting. And the correct answer is, boom, it is D. So this patient most likely has Creutzfeldt-Jacobs disease. The biggest clue here was that he has been working with cadavers. He's been working with human biological material, and he cut himself. So that could be the cause of transmission of those prions. And now he's presenting with mood changes, with minor loss of memory. It's a very bad thing. So it is a misfolded form of a normal protein because that's what prions are. The next question. You have a patient who has a midlife onset of the following symptoms: abnormal, involuntary, jerky body movements, an unsteady gait, personality changes, and chewing and swallowing difficulty, which has led to gradual weight loss. The patient's father has similar symptoms before his death at the age of 45. Cellular analysis indicated precipitated proteins in the nucleus. The disease has its origin in which of the following biochemical problems? Is it A, an exonic deletion? Is it B, a polyglutamine tract in the exon of a defective gene? Is it C, a nonsense mutation leading to a truncated protein? Or is it D, production of non-stable mRNA leading to reduced protein production? You guys are too smart. Honestly, this is not. I don't like it when everyone gets the answers right. Okay, you know what? Let's make this more fun. Write in chat what do you think he has. Man, I swear to God, bro. Like every year they get smarter and smarter. MashaAllah, obviously. Absolutely correct. It is Huntington's, and it is due to a polyglutamine increase. Now, here's a little fun fact for you guys. Imagine you guys become future neurologists, right? And you may notice this pattern with Huntington's patients, right? The patient may tell you, hey, my dad got sick with Huntington when he was 55, right? And now I'm 45 and I have the symptoms. And then his child gets at 35. The Huntington, one of the very interesting things about this disease, it has something called anticipation. By the way, this is just like fun fact, not in your exam at the moment. But the whole idea of this anticipation is each generation gets the disease earlier and earlier. So the first person in the family may have gotten it when in his 60s, then the next person in his 50s, then 40s and 30s until the effectively like the disease kind of like ends with that one person who never like has kids after. So the age of onset is very important. And that, by the way, concludes this topic. We went through everything on this slide, on this image. We went through the breakdown of protein structure from its primary sequence of amino acids, how the secondary structure is just the formation of these hydrogen bonds within the peptide backbone, giving us either an alpha helix or a beta pleated sheets, and then how the tertiary structure is caused by the formation of these extra bonds within the variable groups, whether they are disulfide bonds, whether they're hydrogen bonds, hydrophobic interactions, ionic bonds, and finally, sometimes we have a quaternary structure, which is basically a combination of multiple polypeptide chains. Went through the process of protein folding and how chaperone proteins help in the process of protein folding. Went through the different diseases associated with DNA mutations like sickle cell and cystic fibrosis. Went through the prion diseases like Creutzfeldt-Jacobs disease. Went through how an abnormal accumulation of like alpha beta pleated sheets can lead to amyloid proteins, which is seen in Alzheimer's disease, in amyloidosis and Parkinson's. It's a wrap up of like everything we've taken. Some like if you have any questions, you can send them now. Someone asked, will we be tested in the material of the midterm in the final? To be honest, I don't know. In my time, and again, I'm basically like ancient. I'm like a pharaoh. In my time, usually the vast majority of the final was post midterm lectures, but you would get the occasional like pre-midterm lectures. So I remember it was like maybe like a 70-30 split or an 80-20 split, but it changes year by year. So your best bet is to probably ask your seniors from the year before you. They would probably give you the most like up-to-date information. Any other questions on anything from this topic? Hope you guys found this session kind of helpful. Sorry for all the goofy internet connection issues. Man, I hate the fact that Zoom exists because I'm such a like an in real life person. I hate doing things, through like, man, I sound like such a boomer at the moment, but I'm sorry. You guys have any questions on anything from first year, or I put like my details here, obviously. So anything from first year, anything on medicine, anything in life, I don't know. You can always hit me up. I like to mentor and teach, or any topic that you guys are like confused about. Just hit me up. Be more than happy to help, inshallah. And I think you're supposed to scan this. I'm not sure. It's a new thing. But yeah. My pleasure, guys. My pleasure.
Sorry to interrupt you. I just wanted to say that everyone please go on and scan this code. It's a feedback form. It take like 1 minute or something. Just fill it out, okay? Thanks.
Am I supposed to scan it too?